Hello everyone, so today we're going to be looking at B-cell gene rearrangements, as well as alternative RNA splicing and antibody structure and actions. So some of the stuff we've gone over, but we're just going to go into a little more detail today. So just an overview, we've seen this figure before, is our stages in B-cell development. So we go from our stem cell, early pro-B-cell, late pro-B-cell, large pre-B-cell, and then our small pre-B-cell, and then we have our naive B-cell, as we'll see. So these are important surface marker proteins. So these are how we identify these different phases in research. We can see here antigen independent. Then we can see heavy chain genes. See our rearrangement occurring. Light chain genes, same thing, and our intracellular proteins. So B-cell development stages are marked by the rearrangement of the immunoglobulin genes. The first rearrangements are early in the pro-B cells, where we have DH and GH gene segment joining, forming our late pro-B cells. In late pro-B cells, we have VH to DJH joining occurs. Then we have pre-B cell expressing the mu heavy chain. As we said, mu is associated with IgM. So pre-B cells rearrange these light chain genes, the VJ joining. Then we have our immature cell with the light mu heavy chain on the cell surface as IgM. We can see that here. Mature B cells express both IgM and IgD on the cell surface, and they're prepared from alternatively spliced heavy chain transcripts. B cells that do not successfully rearrange heavy and light chains die by apoptosis. And remember, we have our kappa and lambda. So this is that figure continued essentially. Now we have our immature B cell with our I expressing our IgM. Then we have our mature naive B cell, expressing both IgD and IgM. Then we have our lymphoblasts and memory B cells, which we can see here as isotypes. There's isotype switch using C gamma, which is through IgG. And then we also have our plasmablast cells. And yeah, so these are our effector molecules. And here's our memory cell. So again, our markers of differentiation, so we can identify them. And here we can see VGA rearranged, somatic carbon mutation, and our VGA rearranged again. So as we discussed, there's different constant regions associated with the different isotypes. So we have CMU, IgM, delta D, gamma G, epsilon E, and alpha is A. The different classes of antibodies are produced at different stages of B cell maturation and differentiation. Gene rearrangements allow B cells to switch from production of one class or isotype to another without changing the antigen specificity. So even though we change, we still have the specific binding for that exact antigen or epitope. B cells produce IgG, IgA, and IgE only after an encounter with an antigen through a process of class switching. So these three are not initially produced. So as we saw in the other figure, we have our mu and delta. So we only have our IgM and IgD expressed on the immature naive B cell. And then when class switching occurs, we can switch to these. And we'll discuss how certain cytokines and certain pathogens can elicit each of these. And we can have specified responses. And class switching usually requires help from T helper cells, as I just mentioned. So here are the isotype properties. So we can see here, these are immunoglobulin types. So we got four types of IgG. We got one IgM, two IgA, IgD, IgE. So here we can we got the coding here, what the heavy chain, what's the constant chain, molecular weight, serum level, and half-life. So here are some important features here, which is good to look at. It's like, which one is involved most in the classical pathway? So we can see here that IG3 and IgM. So complement, so just to point out, IgM is one of the most potent immunoglobulin in complement activation. So we can see one thing that's unique is that placental transfer. We have placental transfer of IgG. So this is responsible for the fetus's immunity for quite some time. And here we can see high affinity binding to mast cells and basophils. We can see IgE, which are involved in parasites. Yeah, just keep this in mind. It's a good graph to go back to. So as a result of this different class of antibodies, they are specialized for different biological activities, as we hinted at. So in classical complement fixation, we have IgGM, which is the strongest, then IgG3, then 1 and 2. So for Fc receptor binding on macrophages, IgA is the most potent, We also have IgG1 and IgG3. And IgE, it binds to Fc receptors on mast cells and basophils, as we can see here. And crossing placenta is IgG. So again, here's another way to look at the stages of the B cell. So B cell development stages are marked by rearrangement of the immunoglobulin genes. So we already went over this. So as we went, we can go from our stem cell, early pro, later pro, large pre. Again, this is where our pre-B development of our receptor, small pre-B envelops again and then expresses the IgM. And then our mature B cell, I guess essentially immature naive B cell, has IgD and IgM. And here we can see IgD and IgM are made from alternatively spliced heavy chain transcripts. So now we're going to go through each stage in a little more detail. So gene rearrangement and immunoglobulin production by B cells at different developmental stages. So we have a pro B cell, which initiates gene rearrangements, but no proteins are expressed. So our rearrangement occurs, but here, no functional mu is expressed. In the pre-B cell, we produce the mu-H or mu-heavy chain, which is the pre-B cell receptor. So this mu-heavy chain is known as the pre-B cell receptor, so that's important to remember. And this is on the surface with a surrogate light chain. This is the true light chain gene, so the actual true light gene rearrangement follows. So this is not the fully rearranged gene that's being expressed. So here is our pre-B receptor. And as we mentioned before, all B cell receptors have an Ig beta and Ig alpha receptor, co-receptor. So these immature B cells express IgM on their surface, and mature naive B cells express both IgM and IgD, as we said. So here, we have early pro B cell with the VH to DJH rearrangements, which occur in the late pro B cell. Then we have stop heavy chain gene rearrangement, or progression to light chain gene rearrangement in the small pre B cell. And then we have stop light chain gene rearrangement, and as we'll continue down. So the different constant genes encode for different classes of immunoglobulin heavy chains. The C mu gene is the closest to the J gene, so it's almost kind of goes in the first order. And these are why they always end up on the immature, but here we can rearrange to these later. So it's closest to the J gene segment, which is right here. And so a mu-H chain transcript is produced first. The C delta gene is next in line, and this transcript is allowing expression of IgM and IgD on the surface of naive mature B cells. Class switching, or isotype switching, occurs after B cells encounter an antigen, and this usually involves help from T helper cells. So then gene rearrangement occurs, and the expression of either C constant H chain genes results in the production of different classes of antibody. As we discussed, there's G, which is gamma, and then we have our, which are the 4 subclasses, A, which is alpha, E, and epsilon. So co-expression of IgM and IgD on the cell surface is regulated at the level of RNA processing. So in a naive mature B cell, both C mu and C delta, so IgM and IgD, exons are transcribed in a long primary transcript. This is processed by cleavage and polyadenylation and splicing. So cleavage and polyadenylation at the C mu site, see we can see at PA1, so this is our polyadenylation 1, and exon splicing. So then we have our mRNA encoding the mu heavy chain, as you can see here, after it's been spliced. Then we have cleavage and polyadenylation of the C delta, which is PA2, and the exon splicing to remove the C mu exon, and mRNA encoding the C delta, delta H chain, sorry. So here we can see, now we spliced out this section, and now we have our C delta. Sorry, here's our PA2. So again, here's just another look at this figure. This is kind of the figure we'll come back to as we move down the stages of development. So after RNA splicing, after this occurs with contact with antigen, we can have transmembrane and secreted immunoglobulin molecules. So after encountering with antigen, B cells produce both surface IgM and secreted IgM. And as I said, they're pretty much essentially the same, but here we have this transmembrane region, which we'll discuss. So transmembrane and secreted IgM are derived from the same heavy chain gene by alternative RNA processing. Each heavy chain and constant region has exons corresponding to heavy chain domains, plus 2 transmembrane exons, So the MC is the membrane coating, and a secretion sequence, or SC, which is secretion coating. So see, when we have the SC coded into the DNA here, the RNA, the mRNA, we can see that we have a secreted coating. So we have a antibody that is released here. When we have the MC, when that is used, we have a membrane coating. So therefore we have a hydrophobic transmembrane region and a cytoplasmic tail. And this is the transmembrane form of immunoglobulin. So with SC, this encodes a hydrophilic carboxy terminus, which you can see here. And the SC sequence is continuous with the last constant domain, C domain, sorry. So each H chain gene has 2 potential polyadenylation sites, as we said, PAs and PAM. So here we can see a PAM, PAS, sorry. Here are MCs. They're labeled here as M1, M2, but that is the MC complex here. As you see, both got transferred. So how is this transmembrane form produced? So the heavy chain transcript is cleaved and polyadenylated at the second site, PAM. So here's PAM. The transcript is spliced, so the SC sequence is removed. So this causes the SC sequence to be removed, allowing for MC to work. So then the MC exon joins with the CMU4 exon, and the transmembrane form of the heavy chain is formed, and then we have IgM expressed on B cell surface. For the transmembrane form, transcript splicing is between CMU4 exon and the SC sequence and the 5' end of the MC exon. Production of the transmembrane form of IgM. So we have transmembrane immunoglobulin heavy chains are anchored in the cell membrane by a hydrophobic transmembrane domain at the carboxy terminus, about 25 amino acids long. So here's that carboxy terminus. So again, we have our MC portion coming down instead of our SC, which got cleaved, and now we form the transmembrane antibody, or B cell receptor. So secreted form. So the heavy chain transcript is cleaved and polyadenylated at the first site this time, at the PAS. The MC sequence is removed. So instead of the SC being removed, now we have the MC being removed. This transcript encodes secreted form of heavy chain, and the IgM is secreted by the B cell. So for production of the secreted form, the MC exons are removed. So that's very important. The SC encodes the carboxy terminus of the secreted form. We can see that here. And this results in secreted antibody molecules. The SC sequence is contiguous with the last C domain exon for all classes except IgD. So in IgD, the SC is a separate exon. Production of the secreted form of IgM, so the carboxy terminus of the secreted Ig heavy chains is hydrophilic compared to the hydrophobic in the transmembrane version, and this hydrophilic secretory tail. So in antibody secreting plasma cells, most heavy chain transcripts are spliced to produce the secreted form. So, different forms of IgM. So IgM, we have a monomeric IgM, as we can see here, and then a pentameric. So the pentameric is very good at binding to certain cells, and here's an electron micrograph. This kind of looks like a flat dish shape, as you can see. So these are the transmembrane forms of immunoglobulins or monomers. So this would be the transmembrane. IgM is secreted as a pentamer, though, and is made up of many IgM monomers. So it's basically just a bunch of these put together. 5. And this allows it to bind to so many different antigens. Well, the same antigen, but many at the same time. And these are held together by a joining chain, or the J chain, we can see here, and disulfide bonds, which you can see here. So IgM and IgA can also form polymers. So IgA is the major mucosal antibody isotype, but both IgM and IgA are present in mucosal locations. So these are our most important ones at mucosal sites. So IgM and IgA are often synthesized as multimers in association with the J chain. So again, here we can see our J chains. IgA usually forms dimers, where you can see here there's 2 dimers, which are 2 monomer units of IgA are linked by disulfide bonds. Again, you see those disulfide bonds to each other and to the J chain. where IgM is usually forms pentamers when secreted, and the monomer units of IgM are linked by disulfide bonds again to each other and to the J chain. And IgM can sometimes be a hexamer. So now we're going to look at B cell rearrangements after contact with antigen and look at class type switching. So we've seen this figure before. Here just summarizes all the different types. And then again, here's our heavy chain coding and our half-life. So this is important in kind of our immune response, which is quite important. But yeah, gene rearrangements allow B cells to switch from production of one class, which starts at IgG, which starts with IgM and IgD on the cell, and then will switch with the help of T cells and once it binds to antigen. So class switching occurs in B cells after encounter with the antigen. T cell help is required through T helper cells. So how did they change their antibody class? So the class of antibodies determined by which constant region gene is used to encode the heavy chain. So as we said, there's our mu, delta, gamma, 1, 2, 3, 4, and our alpha and epsilon. So, and this causes rearrangement under the influence of T cells, helper T cells. So we have a switch region. So we can see these S, these are switch regions. So these switch regions are at the 5' end of each heavy chain constant region, and these control the VDJ associations with different constant regions. We receive signals from our T helper cells through cytokines, which causes G rearrangements, and the intervening sequences are spliced out, and there's no return. So once it switches class, it's not going back. So here's cell, we have a B cell after DNA after class switch. So we can hear it's spliced. Now we have delta and alpha. Now we set it spliced again. Now we're left with our delta. Here you see we dealt gamma 1. Sorry, I meant gamma. And then now we see our mRNA for the gamma heavy chain. So now we have IgG. So this is the mechanism of class switching. So B cell switching from IgM to IgG1. So cytokines direct class switching. So like we've learned, interferon gamma can stimulate IgG2 production, while IL-4 can stimulate IgG1 and going to IgE. So this is usually released when we're dealing with parasites, and also involved in some allergic reactions like asthma and stuff like that. So we receive switch signals, which are repetitive DNA sequences that guide class switching. These are found upstream of each immunoglobulin constant gene region, except for C delta. So in switching, recombination between switch signals occurs due to the repair of double-stranded breaks, and the intervening DNA is then deleted. So here we have a break, and then it's deleted. The initial switching from S mu to switching, so here's our S, that's the switching, results in switching to other isotypes that can follow. So class switching involves recombination between specific switch regions, which are those S, here switch regions denoted as S, here again. So that's our switch region, and then sliced, then I'll get sliced off. And then in the constant region, genes. So again, S mu is the mu switch region, so the IgM switch region. So class switching needs some important proteins or enzymes, and here we'll look at AID. So switch regions at the 5' ends of the heavy chain constant genes allows recombination between switch regions and the loss of the intervening DNA. So activity of a special enzyme called activation-induced cytidine deaminase, or AID, is essential in this process. So AID targets switch regions, as we discussed. Here's our switch regions. And this induces DNA nicking, which results in recombination and looping out of the intervening DNA. So for example, we have switching from IgM and IgD to IgG1 region shown here. So this is what's seen in this process. So we have transcription of C gamma gene, and the switch gamma 1 region begins. AID then targets the cytidines in the S mu 1 and S gamma 1. This changes it to uracil and induces recombination process. So essentially swaps out the normal DNA transcript for a mu region, for uracil, sorry, and this results in recombination, and this B cell will switch to IgG1. And can it switch back? We said no, it's not reversible. So again, we have our IgM and IgD are produced, so this is our standard B cell. Then we have AID, selectively targets the S mu and S gamma. So here we see Here's AID acting on the switch regions. Then the DNA and the S mu and S gamma switch regions and nicked on both ends of the strand. Then we have the looping out, as we see, it loops out, then it's excised and removed. And then we are left with our C gamma one, which is our IgG one. So the action of AID. So again, AID is activation-induced cytidine deaminase. It's essential. It is produced only in proliferating B cells and acts on single-stranded DNA in switch regions. During transcription, this deaminase, hence its name, cytidine into uracil. So as we know, we have our DNA, C, G, T. That's not the correct order, but anyways, so we have our C's changed to U, as we can see here. So the conversion of the cytidine to uracil, or uridine, so we see here AID acts on cytidine ring, leading to the deamination of cytidine to form uridine. Conversion of C to U induces DNA repair, since this is considered an error in a normal translation at the switch regions, and these repair enzymes convert uracil to bases in DNA, so it converts it back essentially, and this causes further recombination, so essentially splicing out the mutated, purposely changed areas. So we have our AID, which introduces mutations into the genes transcribed in B cells. AID is involved in class switching and somatic hypermutation, which we'll look at. So AID induces DNA repair, like we said, causes DNA lesions, and results in the activity of repair enzymes, which convert uracil to bases in our DNA. So these different repair pathways can be induced and result in different outcomes for the B cell. But importantly, class switching requires base excision repair and somatic hypermutation, at least in the variable region genes, which can be due to mismatch repair or base excision. AID initiates mutations in different B cell rearrangement processes. So there's class switching, somatic hypermutation, and gene conversion, which is a process used in some animals to generate antibody repertoire diversity using pseudogenes. So here's a kind of a little flow chart here of the different pathways. So take a look if you'd like. So again, AID is produced by proliferating B cells and acts only on single-stranded DNA and only has access to switch region during transcription and deaminates the C to U, as we said. So here, DNA in the variable region in the switch region. Transcription produces the local single-stranded DNA as we normally have in our DNA process. So in B cells, we'll have AID, which attacks cytidine, in the single-stranded DNA to produce uridine. So it swaps out. Now we have a mutation. So now we have a protein called UNG, which is uracil DNA glycosylase, which removes uracil from the aperidic residue. And then this area will be repaired and then it gets excised. So here, again, we see UNG, which removes uracil from the aperidic residue. Then we have an enzyme called APE, or APE1, which cuts the backbone next to the A basic site to form a single-stranded nick. And this single-stranded nick is seen in the DNA, right there. So as again, uracil DNA glycosylase, or UNG, removes uracil. And this is so now there's a nucleotide without a base remains. Then we have endonuclease APE1, which excises the defective nucleotide, causes a nick in the DNA strand. So these nicks in the sequences facilitate recombination. So now we need this error, we need to fix it. And this results in looping out and excision of the intervening genes, as those loops we saw earlier, where it comes out and then it gets sliced. So in this example, we have class switching to IgE is occurring. So remember that involves epsilon. So you can follow the epsilon logos. We see here, epsilon. So we have transcription of the C epsilon gene, and the switch epsilon is initiated by cytokine-induced promoter activation. So we have single-stranded DNA available for AID. AID will target cytidine residues in switch regions, changing it to uridine. Then we have our UNG, which are uracil DNA glycosylase, which removes uracil. This results in a nucleotide without a base, which then remains. Then we have our endonuclease APE1, which excises the defective nucleotide, causing that nick in the strand. Then we have activity of high density. This activity generates high density of single-stranded nicks and double-stranded breaks. And then we'll have our repair proteins, including double-strand break repair protein, which then act. Class switching enables the same assembled variable heavy exon to be associated with different constant heavy genes in the course of an immune response. So again, we have our transcription through the switch region. So our switch region is initiated by the activation of the upstream promoter. We have AID, UNG, and APE1, which introduce clustered NICs. So here we can see our NICs on both strands of the DNA. So we can see it's not just only the one strand. Here we can see our UNG. So repair proteins then act on the double-stranded breaks. And again, we had our double-stranded break repair protein. So then we have repair proteins, which we said were DSBR. These join mu in the C epsilon switch regions, and they are ligated together. The intervening DNA with the constant gene regions is looped out and excised, as we've seen here many times. The result is a B cell that now can produce transcripts for epsilon heavy chains and IgE production. So here we have the repair proteins which initiate double-stranded break repair. You can see that here, fixing the NICs. Double-stranded break repair machinery joins the two switch regions and excises the intervening sequences. So here we can see them, they join the sequences together, the two switch regions. And the selected constant region is now located adjacent to the VDJ region. So now we're going to take a look at somatic hypermutation. So AID is also involved in the process other than class switching. So again, it also induces DNA repair. And there are different pathways which induce different outcomes for the B cell. So we have our class switching, which requires base excision repair, but also somatic hypermutation, which can be due to mismatch repair or base excision. So there's affinity maturation and somatic hypermutation. So B cells proliferate due to activation of antigen. So rapid hypermutation of the immunoglobulin variable region genes occurs, and this B cell receptor a varied affinity for antigen. So here's essentially a nice figure of affinity maturation. See here we have a proliferation, mutation of antibodies. B cells with increased affinity will be chosen to develop further, and this results in stronger binding. So these ones that have better activation by this certain antigen will be the ones that are cloned and produced in more detail and will actually be our memory cells, the strongest. So essentially it's a form of natural selection. Through this, what's the most powerful, I guess you can say, will survive and be proliferate. So somatic hypermutation, B cells compete with each other for presented antigens on follicular dendritic cells, which we'll discuss later. Here you can see that. Those with the highest affinity, like we said, win. So recent positives, they receive positive signals or survival signals, while the others receive apoptotic signals, and then these go on to mature further. The antibody produced will be higher affinity, so this is our affinity maturation. And the remainder of the B cells die by apoptosis. So affinity maturation is due to the random point mutations in immunoglobulin variable domain exons, again through somatic hypermutation, followed by selection of B cells with immunoglobulin mutations that result in increased affinity for antigen. So again, here's kind of like a funnel. A lot go in, and then only one was chosen. So this one had the strongest affinity, and therefore it survives, receives the survival signals, and this is the one that will bind to our follicular dendritic cells and then develop. So this figure shows the distribution point of mutations accumulating after affinity maturation. So the arrowheads, so we see here, these are the arrowheads, indicate locations on point mutations after primary, secondary, tertiary encounter with an antigen. So how many times? So first response, second response, third response. So we can see here all the different mutations that are occurring, even if we've already become immune to this thing. So this is say we've got a vaccine, now we've got our memory response ready. So even after we have the secondary response, so we catch it again, our system will still improve and go through the same process of somatic hypermutation and affinity maturation, as we see here, and keep developing. So the more and more this keeps going on and as we get reinfected with that virus or whatever. So we keep developing stronger and stronger immune response. But again, note that point mutations are mainly in the complementary determining regions of the variable domains. So this figure also shows the distribution of point mutations accumulating after affinity maturation. The point mutations are mainly in the CDRs, like we said, of the variable domains. Mutations in CDRs are selected for since they are most likely to result in an increase in antigen binding affinity. So the B cells with the optimal mutations in the CDRs are the ones that receive positive signals to survive. So again, as we have our responses, ones that bind strongest survive, then the ones that were, I guess they could be used to be strong, but no longer are in comparison, die off. So here we see red, just so you know, is locations of point mutations. So the selection of immunoglobulin molecules with improved antigen binding sites occurs during an immune response. So 1 week after a primary immunization, such as a vaccine, we have IgM production, which predominates. Then we have some new sequence variations in the variable regions, mainly in CDR3. So 2 weeks after immunization, we have IgG is also made. Both IgM and IgG show increased variation in all 6 CDRs. So we can see that here. As response continues, we switch from IgG. So we have somatic heart mutation, and through affinity maturation, we have higher affinity than the original IgM. So here's just a summary. As we talked about the differences between our T cells and B cells. So B cells, we have all these processes. So somatic heart mutation, and it was secreted, where T cells we do not. But like we discussed, our actual diversity is similar. So summary, antibody diversity. The coding and assembly of antibody molecules randomly chosen variable diversity and joining, so our V, D, and J, combine to form RNA transcripts, which then is translated into a protein chain. 2 heavy and 2 light chains are assembled into an antibody molecule, forming a constant region and a variable region. These gene rearrangements in B cells are used to generate a wide repertoire of antibody from a limited number of gene segments. Gene rearrangements in the class switching process, after our encounter with the antigen, allows the B cells to change its Fc region, like we said, into other types of antibodies, which allows for greater antigenic specificity. But this isn't altered. So again, antigenic specificity isn't altered, so it's always the same antigen we're attacking, but increases its strength. Further improvement of antigen recognition ability can occur through somatic hypermutation and affinity maturation, which are very important processes. So here we see our primary immunoglobulin G, or M, sorry. Kind of get a visual here in the red sections. Somatic hypermutation and the class switch. So the primary antibody repertoire is diversified by processes that then modify the rearranged immunoglobulin gene. Somatic hypermutation leads to changes in the variable region, and class switching leads to changes in the constant region. And these genes are used to generate immunoglobulin molecules. So constant region allows us to have our different types, and this will increase our affinity to the antigen. So now we're going to be a closer look at antibody structure and function. So we've talked about seen this figure before. Antibodies play a dual role. They can recognize and attach specific antigens through the role in variable regions. They can also neutralize or destroy antigens through biological interaction through the constant regions. And the antibody structure is ideally suited for these dual tasks. It enables precise specificity for antigens and conserves structures for biological activities. So again, is involved in ADCC and also involved in complement. See here the classical pathway. So the antibody structure enables them to recognize and bind antigen with the variable region and interact with different cells and remove or activate the antigen, and is involved in the constant region. So this Fc constant region. So that's our constant region here. The stalk of the Y. So that's involved. That's where this binding occurs. So the main functions of antibodies, as we discussed before, is opsonization, neutralization, and complement fixation. So here you can kind of follow the 3 different roles and see, kind of depict the different functions here. So complement activation, lysis and ingestion. Opsonization makes it easier for macrophages to see it and they phagocytose them. And neutralization, it causes ingestion by macrophages, so it kind of inactivates the virus and makes it exposed to phagocytosis. So complement fixation. So this is mainly involved in the classical pathway of complement activation, is initiated by the binding of the C1q, so the C1q complex, to an antibody on a pathogen surface. and IgM is very efficient at complement fixation. So IgM is basically the best. So here we had a pentameric form, so combined to so many different complements at once, so it causes really strong binding. Most IgG can fix the complement as well, but at least 2 IgG molecules are required. So here we see we need 2 molecules, where 1 IgM can bind all at the same time. We need these 2 molecules of IgG at once so they have the complement, or C1q, bind properly to at least 2 immunoglobulin molecules. And both of these activates C1R, which cleaves and activates the serine protease C1s. Neutralization. So neutralization of bacterial toxins by antibodies protects cells from damage. So antibodies can also prevent attachment of bacteria to cell surfaces and block virus infection of cells. So IgG, A, and M can all neutralize, but high affinity antibodies best. So essentially, let's just say there's a bacteria in the blood. It can basically get bound by free antibody or other antibody on molecules. Essentially blocks its function. So for example, like we said here in the virus, these antibodies bind to the virus particle that was released, let's say released from another cell. Basically gets clumped up with a bunch of antibodies here and essentially cannot enter the cell to reproduce in the next cell. So here we have our initial response. Now we have our adaptive immunity. So new virus is produced. This cell just literally just can't get in. So we can have neutralized antibodies. And a lot of these are seen with IgA in our mucus. As we inhale certain things we've exposed to before, we can have our antibodies block it so we don't even get infected. So affinity of the antibody for antigen determines the strength of binding. It reflects the ability of the antigen to stably associate with the antibody. Antibodies specific for the same epitope can differ in binding affinity. The average affinity of antibodies produced by a B cell usually increases over time, as we said, with affinity maturation. Antibodies made during the secondary response have higher affinity than those made in the primary response. So IgG and IgA are higher affinity than IgM. This is why this happens in our class switching and our affinity maturation. So you can see here, IgM is the first antibody made against the pathogen through somatic hypermutation and affinity maturation. We have the switching of our antibody isotypes to IgG, which allows delivery of pathogen to phagocytes with a much stronger binding. So as we discussed, we have antibody-dependent cell-mediated cytotoxicity. So antibodies coded. So here's a figure if you just want to keep watching while I speak. So antibody-coded target cells can be killed by NK cells and ADCC. NK cells have Fc gamma receptor 3 on their surface. So when they encounter cells coated with IgG, the antibody, they attach and kill the target. So certain cells tagged by antibody, NK cells will come in and attach and release their granzymes and perforins and kill the virus infected cell. So again, here's the animation of the NK cell killing an infected cell, which antibody molecules have attached through ADCC. And ADCC can also be mediated by macrophages. So it doesn't have just be NK cell, macrophages can come in and kill the cell as well. So the Fab portion of the antibody binds to the epitope, which you see here. Well, the Fab portion, that's the Fab portion of the epitope. And the NK cell binds to the Fc receptor here. And yet again, as the receptor on the NK cell, the NK cell gets close to the target and then releases its perforin and granzymes, causing apoptosis. So here's another great figure about the functions of each class of antibody. So again, here's our class. Strongest at neutralization, we can see here, is our IgGs and our IgA. Opsonization, we discussed, was IgG1, IgG3. Activates complement system, as we mentioned, IgM was the strongest, along with IgG3. And sensitization of mast cells, which is very important in IgE-mediated responses. So these are the human immunoglobulin classes. Compared, we have the mice, which is down here. Mice have 4 IgG subclasses, but they are IgG1, 2A, 2B, and IgG3. IgD is a surface receptor on all mature IgM-positive B cells as part of their B cell receptor. My mistake, this is just other variables if you want to look at them. So what transports across the epithelium the most? IgA, as we discussed. IgA is pretty much the most present in all epithelial tissues. The placenta, we mentioned this before, is IgG. Extravascular sites, we have our IgGs in 1 to 4. and mean serum level. So what's the most present? You can take a look here. So IgM. So IgM is the first class produced in immune response to an antigen. It accounts for 5 to 10% of serum immunoglobulin. It serves as the surface immunoglobulin for B cell receptors. As we said, IgM and IgD are expressed first. It's on our primary B cells. It's secreted as a pentamer. Here we can see. It has 10 antigen binding sites per IgM. So 1, 2, 3, 10. And it's very efficient at activation of complement cascade, leading to opsonization. IgG. So IgG is the most abundant class in the serum. It accounts for 80% of serum antibodies. And there are 4 subclasses, as we've seen, 1, 2, 3, and 4 in humans. And the roles in addition, it has also roles in addition to neutralization. It is involved in complement fixation, but not as good as IgM, with IgG 1 and 3. It is also involved in opsonization through the Fc receptor. There's the Fc receptor of IgG1 and IgG3. And also can cross the placenta and help with protection of the fetus. So with the IgG, there are 4 subclasses, and these loops show domains stabilized by intradomain disulfide bonds. And we can see here the variable domains of the L chains are shown in amber, and the variable domains are shown in pink. So IgA. So here's a picture. So just so you know, dimeric IgA is the major class in the gut lumen. IgA is transported into the gut lumen by intestinal epithelial cells, which we discussed in our lymphatic system stuff. We talked about the MALT. IgA is produced by plasma cells in the lamina propria of the stomach and the intestines. It's the key secretory antibody involved in defense of mucosal surfaces, so our MALT. Usually secreted from B cells as a dimer in plus J chain. The J chain has a role in transport across the mucosal surfaces. So this J chain binds to the polymeric IgR, so here we can see polymeric immunoglobulin receptor, on the intraepithelial intestinal epithelial cells at the basolateral surface. This is endocytosed, so we can see here, endocytosed, and transported in the vesicles to the apical surface of the gut lumen, which you can see up here. This enzymatic cleavage of the poly-IgR, these fragments stay attached as IgA as a secretory component, and this improves resistance to proteases. So essentially, we saw that IgA is dimeric, so it releases that dimeric IgA, and these will bind to different bacterial toxins in our stomach. And here are the roles of IgA. We can say IgA neutralizes pathogens at mucosal surfaces, so both our MALT and in our airways, and it's also secreted into breast milk, and also helps with protection of infants. So again, IgG is secreted across the placenta, while breastfeeding releases IgA. So IgE. As we've hinted to before, IgE is involved in some allergies, but it's also important in parasites. So IgE is found mainly associated with mast cells in tissues and basophils in the blood. This is via the Fc receptor for IgE, which is Fc epsilon receptor 1, which is very low serum levels. We also have very low basophil levels in comparison to other cells, so it makes sense. Immunoglobulin binding to the allergen will result in cross-linking, and this will result in mast cell and basophil degranulation, and these release their granule mediators, which include histamine, which can result in symptoms in allergies, asthma, and anaphylaxis. So essentially during these reactions, but we'll have whole lectures on different immune conditions, but these allergens will unnecessarily stimulate these IgE molecules, and they'll stimulate excess amounts of basophils and mast cells, causing too much histamine release. And again, this is where we get our crazy symptoms that are not fun. But IgE is not just a bad guy. It's very important in the defense against certain parasites, where degranulation may provide host protection. So here's our summary. So antibody molecules play a dual role. They have a role in recognizing and attaching to specific antigens through their variable regions. They're also very important in neutralizing or destroying antigens through biologic interactions, through their constant regions with their different isotypes. The antibody structure is ideally suited for these dual tasks. It enables precise specificity for antigens and conserves structures for biological activities in our Fc regions, so a lot of stuff can bind to it, but also be very specific. And the antibody has several key actions and roles. Again, neutralization, opsonization, complement fixation, ADCC, and mast cell degranulation. And there are different classes with our isotypes of antibody, which can vary in biological activities, as we discussed. So again, hope you guys enjoyed that one. That was a longer one, but you guys can see that antibodies and all the different classes are super important and crucial to our immune defenses. Take care.